Very good. I know some people still grabbing their food and logging in, but wanted to start getting started at least informally so we can spend some good time. Very privileged to have one of our own faculty, Olivier Gevaert. You've been here before me, so plenty I've been learning from you, and I wish I could learn more. I've only heard your last talk, I think, a year ago. And so very excited to hear more about that. Before we dive in, though, tell us more about yourself. Where were you born? Where'd you grow up? What is that accent? And how did you arrive here?
I expected that question and it's prepared.
Oh, he's ready for it. So predictable.
So I'm originally from Belgium, and I did all my training there. I actually my primary master, or whatever you call it, is industrial engineering. So I studied in a college in Ghent, in Belgium. I also did a Erasmus study in Germany for 4 months at the end of that. And then I actually did a master after master program. It existed back then, it doesn't exist anymore, actually, these programs in Belgium. In Leuven, the other sort of major city in Belgium with a university. And actually that was sort of how I got excited about AI. I was very excited about computer vision, text mining, and things like that.
What year was that at the time? Because it was maybe
2003.
Yeah.
So that's 2003. And that's how I learned about the concept of a PhD. I never heard of this before. I didn't know what it was. But then some of my colleagues, students in that program were thinking of doing a PhD. I was like, okay, maybe that's the next step. So that's what happened. And then I joined the lab of Bart de Moor in Leuven in Belgium. This was in the Department of Electrical Engineering. And there was a bioinformatics group in Leuven there. I was co-advised by Dip Timmerman, who is the middle person here. And he is a gynecologist. And then I also had a mentor, Frank de Smet. And so actually I then was pitched a project on multi-modal modeling of clinical data, RNA sequencing, and imaging data of ovarian cancer using Bayesian networks, Bayesian graphical modeling. So that was not, I didn't know anything of this area or at all or anything like that, but that was what I was offered, so I said yes. And that's sort of how I got started in bioinformatics, and I learned sort of a lot of medicine and molecular biology just by reading a lot and learning from my peers. Maybe one interesting thing about my advisor, Bart de Moor, so his main focus is mathematics and mathematical engineering, and he's very fond of this mathematics genealogy project, which is sort of, it keeps track of the genealogy of mathematicians. And so he made sure that all the students submit their PhD to this website. So I was number 55. I was his 55th PhD student. So he has 89 students and more than 150 descendants. And so he traces back to famous mathematicians in the past, including Newcomb and Kalman, Gauss, and then also French mathematicians in the past. So this was my PhD thesis in 2008. So I developed a framework using Bayesian modeling, Bayesian graphical modeling. So this was actually at the time, so when I started my PhD, neural networks were really out of fashion. So when you couldn't get any funding with neural networks, so it was just at the end of sort of like before there were sort of still a lot of activity, but at that time it was definitely not popular. So that's why I focused on Bayesian modeling and Bayesian graphical modeling. And then I joined Stanford in 2010. I came to the radiology department with funding from the Belgian American Educational Foundation and also from the Fund for Scientific Research in Flanders. And I had just randomly connected with Sylvia that time. And she said, I asked her, can I join your lab as a postdoc? I have my own funding. And she said, yes. And that's how I joined her lab. And the reason I joined her lab was she pitched a project which was radiogenomics, sort of had done a lot of work in genomics and bioinformatics, and was very interested in like bringing radiographic images and sort of CT and MRI images. And that's why I was very happy to join her lab. And I stayed in the lab until 2015, first as a postdoc and then as staff. And then I joined BMIR in 2015 as assistant professor and then associate professor since 2022. And that's sort of the history.
Loaded question. Why did you join BMIR as opposed to radiology or any other department?
I thought about that. So because I don't, so I'm an engineer, a data scientist, bioinformatician. I don't want to focus on just one data modality. Also, as you've seen, my PhD was about multimodal modeling. So I interviewed in radiology departments as well, but I felt that would be too constrained. So my reason to join BMIR is because it was more general focus on biomedical informatics. That was sort of the main reason.
Any regrets? No, don't answer. Of course not. Well, we're very glad to have it, Dr. Gevaert here. Go ahead, let's dive in and hear about all of the different modalities. Go for it.
So I hope I don't have to convince you that machine learning has had a major influence on biomedical data analysis across many different types of data, ranging from clinical data, molecular and imaging data. Many studies have shown that you can discover biomarkers, develop decision support models from all these different types of data in the context of precision medicine. And what do I mean with precision medicine? I mean optimizing treatment of patients or a subset of patients using biomarkers. There are several examples already in use right now. For example, in the field of oncology, there's gene expression assays, for example, to diagnose thyroid nodules, to determine treatment in breast cancer, and to diagnose colorectal cancer based on methylation. Also, imaging biomarkers are emerging, and there's have been recently lots of FDA approvals in the context of imaging, especially radiographic images. And the goal of all these biomarkers is to assign diagnosis and treatments to patients for patients. But high-dimensional multimodal patient data, in this context, each modality has strengths and limitations. So there's also different data forms. So data can come in text, vectors, quantitative features, images, different types of images. So we need machine learning methods and models that can represent all these different types of data. So that's what we focus on in the lab, is how can we develop models for heterogeneous data embeddings, and then how can we handle multimodal fusion, because now we have also access to all these different streams of data. And so we are focused on developing robust methods that can handle all these different types of data, and also missing data, because we can have also lots of missingness. We can also miss data modalities. And we also want to use pre-training and transfer learning. So this is just an overview of sort of an example in the context of cancer patients. So these are sort of four main streams of data that we typically have for cancer patients. We have the electronic health records. Now we have more and more routinely molecular data. We have foundation medicine type assays like somatic mutations of a handful of genes or a few hundred genes. RNA sequencing is becoming more and more routine. We have also digital pathology. These are huge 2D images that we can analyze with machine learning and AI. And we have radiographic images. And the advantage of radiographic images is that they are non-invasive, so we can track also over time. And so our goal is really to develop these frameworks, pipelines, methods to represent all these different types of data in a lower dimension, to combine them and to use them for prediction. So this is what I'm going to try to focus on today. So I'm going to give you an overview of the work we have done in the last couple of years across different topics, and we'll see if we get to the end. So first, one area in the lab that we focused on a lot is in computational epigenomics. So this is more in the context of multi-omics data fusion. So we're bringing multiple omics or molecular data together. And one of the reasons is that DNA methylation has been recognized as an important process in oncology, and hypomethylation of genes has been shown to be important as a mechanism to activate or deactivate genes. For example, MGMT methylation glioblastoma is a very important biomarker, and it determines treatment with chemotherapy. And also BRCA1 methylation in ovarian breast cancer has been shown to be a mechanism to deactivate these genes. So we started already more than 10 years ago to develop methods to identify hyper and hypomethylation. And we developed an algorithm called Metalmix. This is a data mixture model that identifies the methylation states across a population of patients. So here we have the different states. Then we also bring in normal DNA methylation data. So we need to know what is the normal state for a particular tissue in a particular cohort. And then we also bring in expression data. So we link methylation with expression because we are only interested in capturing methylation events, hypo or hypermethylation, when it is correlated with expression. So this is Metalmix, was developed already a long time ago. And we do this for each gene or for each CPG site within a gene. So we've recently, last year, updated this work, and we've developed EpiMix and added a lot of use cases and improved the software. So this is an R package. We add now more linking to common data sets, such as the Cancer Genome Atlas and GEO. Also, before it was really focused on cancer, but the updated workflow is disease agnostic, so you can use it across any disease area. We also add some more support for missing value imputation, batch effect correction. And then we also added a number of use cases. So before, Metalmix was really focused on protein coding genes, and now we also add other modes, such as enhancers, microRNAs and non-coding RNAs. So essentially, this means that we now can run Metalmix in all these different modes and look at other non-coding elements of the genome and look at their hyper or hypomethylation states. Then we also add some downstream analysis, better integration of gene enrichment analysis, subtyping, modes of enrichment analysis, and so on. And we've updated this in an R package. So one major update I want to show you is also this concept of deconvolution. So in many cases, molecular data in oncology is captured of tissues, and tissues are mixtures of cells. And a lot of work has shown that we can actually also deconvolve these expression signatures or methylation signatures across cell types. So we added this to EpiMix, where now we can deconvolve the methylation data and also the expression data across these major cell types: epithelial cells, which are the cancer cells, fibroblasts, lymphocytes, and endothelial cells. So we use two different algorithms. We use CyberSort to deconvolve the expression data, the RNA sequencing data, and we use another algorithm to deconvolve the methylation data. And now this essentially allows us to make these EpiMix calls or states for discovery of these states cell type specific.
Could you just clarify what you mean by deconvolve? I'm not sure what it means, and I'm guessing I'm not the only one.
So we're collecting these molecular technologies, RNA sequencing and methylation from bulk tissue, which means they have a mixture of cell types. So deconvolution is a mathematical way of disentangling these data. And you use some additional data to do this. We can use single cell data or other ways of figuring out what are the percentages of cells for each of these different cell types.
Thank you.
So that allows us now to make these cell type specific calls. And it essentially reduces the amount of noise. And we validate this by looking at a published work from this paper here, Vass et al., where they looked at hyper and hypomethylation in lung cells, which was a very a single cell type. And we see that if we look at the EpiMix calls that are cell type specific, that the overlap is much more significant compared to the calls based on bulk tissue. Essentially, the EpiMix is integrated bioinformatic algorithm to combine DNA methylation and RNA sequencing. And we use it for identification of driver genes, patient subtyping, and also for prognosis. And the main thing that we added now in this recent work is we also extend epimix to non-coding genes, such as non-coding RNAs, microRNAs, and enhancers. Any questions before I go on for the next?
Keep building.
So now let me talk about a different topic in the lab, and I mentioned it already in the beginning, is this problem or challenge that we don't always have multimodal data for all patients. And one area we have been focused on is what we call cross-modal modeling, where we impute one modality from another. And here we're going to look at how we can use digital pathology data. So some modalities are more routinely acquired than others. And for example, molecular data, RNA sequencing is not yet routinely acquired, but it's important for subtyping, for pathway activity modeling, for drug discovery. Whereas digital pathology is the cornerstone for diagnosis of cancer patients, so we have this routinely for all patients. So can the H&E image, can it act as a surrogate of the RNA-seq profile? So that's what we tried to develop. We looked at data of 7 cancers, brain, lung, kidney, breast, and so on. And we developed a model that uses whole slide images. And this model is called Sequoia. It's a vision transformer that essentially uses the image as input and predicts the RNA-seq profile as output. And we developed this model based on Cancer Genome Atlas data and data from GTAX. GTAX is data from healthy tissue. So we essentially have matched RNA sequencing and images from across all these tissues that we have here on the top. And then we also use data from CPTAC, which is another similar TCGA-sized cohort, but also includes proteomic data. And then we had some lung cancer data from Stanford. So the main idea is really to use these data sets and use GTAC for pre-training to develop this model and then predict the RNA-seq profile. And then we do some validation. We look at the genes that we can predict what are their functions using GO and equivalent databases. And we also do some spatial validation because we had for one cohort, for glioblastoma, we had a validation cohort where we had spatial expression of the gene. So we can also look at spatial visualization. So this is the model. So the first step is we use ResNet, which is a way to extract features from batches of these images. Then these batches are clustered into K clusters, and then these clusters are fed into a transformer. And then the output is a prediction of all genes in the human genome. One other important aspect is that we pre-trained this model using GTAX, so these normal tissues. And I will show you the effect of pre-training. And then we also compared with a state-of-the-art model called HE2RNA. So here are the results. Let me walk you through this graph. So on the left side and the y-axis is the correlation coefficient. So we looked at both correlation coefficients and error to evaluate this model. We want high correlation, of course, with ground truth and low error. The black squares are the number of genes we can predict for each cancer. So you can see, for example, for breast cancer, you can predict up to 14,000 genes reliably, versus, for example, lung squamous carcinoma, it's only a few thousand, and we can predict accurately. Also, our model for most cancers performs better than the previous state-of-the-art model. And what we also saw was that for cancers where we had less data, for example, pancreatic cancer, pre-training with GTAX improved the performance. But for example, in breast cancer, pre-training using normal tissues didn't really help. So the more data we have in a particular disease area, the less important pre-training is. So then we also looked at validation. So we had the second cohort, which is CPTAC, across all these tissues. And we were able to validate the genes that we were able to predict based on TCGA. And that's shown here. So you can see how many genes we could validate across these cancers. Then we also looked at some gene enrichment analysis where we wanted to know which genes, which pathways can we actually predict well from these images. And you can see that, for example, T-cell differentiation, cell proliferation, cell cycle are pathways that are very well represented. And then we also focused on lung cancer, and here are some pathways in lung cancer that we could reliably predict from the H&E. And this is important because this could be used, for example, for determining treatment or using gene expression signatures, of which there are many across cancers, that we can use to maybe diagnose or use for treatment or for other purposes. Then, as I mentioned, we also had spatial data in glioblastoma, and we looked at the spatial expression of single genes. I want to remind you that the model was trained on bulk expression, so we are not, the ground truth is really bulk expression for the image, so we don't, this is essentially going beyond what the model was trained for. But we could show that for a number of genes, the ground truth correlated very well with the prediction. So each time I'm showing you here in the left column the prediction of the model, and on the right column is the ground truth from this spatial transcriptomic data set using the 10x platform. So we could show across a number of genes and across a number of cases that there was a nice correlation between the predicted virtual expression across the slide and the ground truth. So to summarize this work, so we developed this vision transformer that allows imputing RNA-seq profiles. We show that it outperforms previous state-of-the-art models. We also did some validation in CPTAC and in the Tempus cohort and some visualization in glioblastoma. And we're continuing working on this model and improving it and also extending it to more tissues. So now, I also want to show you an example of the opposite. So we just used images that are very routinely collected in clinical medicine and that can be used to estimate RNA-seq profiles, which are not yet routinely used in oncology. But now we also wanted to try the opposite. How can we generate synthetic images from RNA-seq profiles? And again, the motivation for doing this work is very similar as what I mentioned before. Often modalities are missing. Some modalities are more expensive to generate than others. For example, spatial transcriptomics can also be time consuming, or maybe we don't have access to certain modalities in publicly available cohorts. And modality imputation can just be useful to complete multimodal data, or to also generate partially synthetic data, and that could have advantages in terms of data sharing. So I'm sure you're all familiar with models like DALL-E 2 or Midjourney, where you can use text to generate an image. So we developed a similar model where instead of text, we use an RNA-seq profile, which is the expression or the activity of all the genes in the human genome, and then use a cascaded diffusion model to generate synthetic images that represent that expression profile. This is motivated by recent work where text-to-image modeling has been very successful. And previous work using GANs has also shown that you can generate images from RNA profiles. And here's our objective. So we wanted to create a multi-cancer model that can generate images from RNA profiles across different tissues. We wanted to have a single architecture, so a single model that can model different types of tissues. And then we want to evaluate these synthetic tiles and show that they can be used for maybe pre-training or to improve downstream tasks. I also wanted to show that these tiles, these synthetic images, can substitute real data, but that they also represent real images. So here are some results. So we essentially developed this cascaded diffusion model, very similar to the DALL-E architecture, trained this on TCGA, and we were able to generate very high quality synthetic images. So at the top, I'm showing you for glioblastoma and lung cancer, lung adenocarcinoma, synthetic and real images. The first thing we tried to do to prove that these synthetic images are useful is look at the cell type distribution. We used an algorithm called HooverNet that essentially detects the major cell types like tumor cells, lymphocytes, connective tissue, and so on. And we show here for lung and for renal cell carcinomas that the proportions of cell types that we detect in each of these images are very similar to real images. Then the next thing that we did is we wanted to show that pre-training on mixtures of real and synthetic data can have the same performance as. Go ahead.
I have a question about the evaluation of the synthetic data. Is there like a set criteria that's used to evaluate whether these images are like similar or close enough to real data that you would be able to use them as a substitute?
There's so I'm going to show a number of ways of proving that these synthetic data are useful. We also calculated some metrics that look at similarities. I'm not going to show that here, but we also did analysis like that using the fresh air metric that calculates essentially the similarity between synthetic images and real images, and we showed that they are very similar. So there's lots of indirect ways. We also have done surveys with pathologists to see if they can distinguish real and synthetic images. And then we're going to show some more sort of indirect ways of proving, but it's a sort of an ongoing effort in trying to see what are the use cases for synthetic data, for sure. Any other questions? So here we trained a model on mixtures of real and synthetic data, ranging from 100% real data to up to 75% synthetic data. And we show that the performance is not different on a real test data set. So this is a model that does tissue classification. So we show that we can train from mixtures of real and synthetic data. Similarly, if we train on synthetic data, we can keep improving the performance. The more data synthetic data we use in pre-training, we saw that increasing up to 5,000 data points, we kept improving the performance in a pre-training scenario. And here is a very specific use case. So this is a prediction of microsatellite instability in colorectal cancer, which is a very interesting area for pharma because they want to have cheap assays to predict this from existing data. And we show here also that if you train a model on synthetic data beforehand, and then we add more and more real data, that the model that was trained on synthetic data keeps outperforming the model on real data.
On good synthetic data. Bad synthetic data would presumably hurt.
We don't select any. We don't use any metric to read out data. Yeah. So we previously did some work using GANs in this area, but we could not train GANs that were multi-tissue. So we followed up that work with this work that I just showed you using a cascaded diffusion model, where we actually were able to develop a model across 5 tissues, and we have now also extended it to 33 cancer types. And we showed that we can that these synthetic data, these tiles mimic real data, and that we can use this in lots of efforts, including pre-training, transfer learning, and so on. Any questions before I move on?
Probably missed it. You surely said it. Where did the synthetic data come from? It must have been trained on something. It has.
So it was trained on TCGA. So the same cohorts that I mentioned in the beginning. So the model was trained on 5 tissues of TCGA. So we have the images and the RNA-seq data. So very similar to the course we used for the
separate from what you call the real data when you do the subject training.
So the model is trained on real data, right? So and then we can start generating images with made-up RNA-seq profiles.
So is it sort of like your data augmentation from a training set you've already got? You just make a synthetic, a million variations based on your own, and it's somehow smoothing out some process that seems to optimize downstream performance.
Yes. So we released a data set of 1 million images that we generated across these 5 tissues, like 200,000 images each. We also try this. We can also sample out of distribution. So we can use our RNA-seq profiles of other tissues that we didn't train. And that seems to, we're still working on that, but because as the question came earlier, it's hard to prove is this synthetic data useful, right? So so we're still working on metrics and ways of evaluating synthetic data.
Yeah, go ahead. But during the generation, synthetic data generation procedure, are cell type involved? I mean, are those purely generated from expression data or?
So that's a very interesting question. So the data that we used is also bulk RNA sequencing. That means that it represents multiple cell types, and that so the images also reflect multiple cell types. But we did some experiments where we also used the same methods as before, deconvolution. We deconvolved the data into different cell types and then generated synthetic images from those cell types, and we see the expected increase or decrease of the of that cell type. Yeah.
I'm not a biological person, so it seems that every cell type has a specific shape, right? Are you considering incorporate cell type shapes and the patterns in the tissues into the model, or?
So actually, that's what I'm showing you here is the use of HoverNet, which is a model that was trained by another group, and that's exactly what they use, right? So they are able to distinguish all these different cell types based on shape, texture, size, and so on, right? So that's exactly, yeah. So there's lots of different ways of analyzing this image data. Great. So now I want to also discuss another project that is really focused on spatial transcriptomics data.
It's supposed to look like a map of the US, sort of squished.
That's a brain tumor tissue. So spatial transcriptomics or spatial omics has been a transformative technology in oncology and also beyond in the last couple of years. It essentially allows us to capture spatial ground truth RNA-seq profiles or even other molecular methodologies for tissues. And it again gets to the cellular heterogeneity and heterogeneity that's often observed in cancer. So we had access to this brain tumor spatial transcriptomics data set of about 25 patients. And we also had the images, the H&E images, and we wanted to investigate whether we could use the imaging as a surrogate for the spatial transcriptomic data. So what I'm showing you here on the left is a representation of these spatial data. And on the right is a clustering of the spatial transcriptomics data. And Eric, a postdoc in the lab, he defines these 5 cell types that we can distinguish in glioblastoma. And this also confirmed previous work from single-cell RNA-seq data in glioblastoma. So you can see these 5 cell types, neuroprogenitor cells, oligodendroprogenitor cells, mesenchymal cells, 2 subtypes, and reactive astrocytes. And we also had normal cells. So we also had data from normal cells. So the first thing that Eric tried was that he wanted to see, can we predict locally the cell type from the H&E image? So again, we have the H&E image. We use a resonance model that represents the features of these patches. And then we predict these 5 cell types plus normal. And as you can see here, we can actually very accurately distinguish the cell type at the patch level from these tumors. Then we used another cohort, which is called IV gap cohort, where we had local ground truth for proteins or using RNA in situ hybridization. And we wanted to use this cohort as a validation of these 5 cell types. And I'm showing you here 2 cases. So these are two patients and their tumors. So on the left, for example, I'm showing 4 genes that are representative of the 4 of the cell types. For example, HIF1 alpha is important in hypoxia. So this is a hypoxia gene, and we show that the prediction of the model for this cell type is very nicely correlated with the staining of that gene. So this is a sort of a validation of this model. So now what we wanted to do is, so spatial transcriptomics is still a very expensive technology. It's a very novel technology. So it hasn't been used on many patients, also because it's cost prohibitive. But because now we can predict these cell types from these H&E images, we can now use this model across, for example, all of TCGA and CPTAC and make predictions of these cell types. And that's what we did. So we imputed all these spatial maps of these cell types. And then we looked how important these cell types are for prediction of prognosis. And here I'm showing you that if reactive astrocytes cluster together, it's very bad for prognosis. So this is the hazard ratio. And this across 2 cohorts, so this TCGA and CPTAC, versus if they cluster with other cell types, in particular with NPCs and with mesenchymal hypoxia cells, then it's actually good for prognosis. And this is true in both cohorts. So this is sort of a way of bringing the richness of spatial technology to larger cohorts using this intermediate model. And then we also looked at, can we actually directly predict prognosis from the images? So we built a model, a ResNet model, that can predict prognosis. And that this essentially gives us an aggressiveness map. So this gives us a visualization of which areas in the tumor that are predictive of prognosis. As you can see here, so red means like very bad for prognosis, where blue means it's very good for prognosis. And then we also overlaid these maps. So this is the prognosis map, this is a cluster map. So now we can also learn which cell types are more bad for prognosis versus others. So to summarize, so we used the spatial cohort to develop a model that can predict cell type, and then we can use these models and deploy them in any cohort that has H&E data. So we can also directly predict prognosis. And we also did validation in the ID-gap cohort and the CPTAC cohort. Any questions before I move on? So now I'm going to switch gears a little bit and show some work that we have done in radiographic images. So we have done in the past 10 years or so, we have done several projects in this area, starting with more traditional computer vision methods and feature extraction such as radiogenomics. Then moving on to deep learning, such as developing convolutional networks and autoencoders. And the main tasks here are segmentation and classification. So the work I want to show you today is focused on lung cancer, where we have lung CTs. So we have these lung CTs of lung cancer patients, and what we want to represent these 3D images of these lung tumors in a lower dimensional space. So Terry in the lab, he developed a variational autoencoder that can sort of represent these images, these 3D images in lower dimensional space, and then we can use them for various downstream tasks. And then this is a very general workflow that we can also use for other organ systems, such as brain MRIs or liver imaging. So Terry used data from a number of cohorts. We used 2 cohorts for pre-training the model. These are cohorts that have both benign and malignant lesions from the LIDC and LMDB. And then we used a Stanford cohort for further fine-tuning and also for developing models for downstream tasks. So here I'm showing you 4 approaches that we are comparing here. First, we want to look at radiomics features. So this is sort of the traditional way of analyzing radiographic image data, where you have a segmentation of a tumor and then you extract features. And then you use these features to build a predictive model. In this case, we want to predict stage, which is essentially a categorical version of size. Then we compare with the convolutional network. This is a supervised model that also predicts stage. Then we have the variational autoencoder that we built, which is unsupervised. And then we also have a combination. So what you can see here is that all these models perform very similarly to each other, with the combination of radiomics features and a variational autoencoder trending to have a better performance. But the main advantage of this is that our variational autoencoder is unsupervised, so we can use it for several tasks. We can also use it for other predictions, such as predicting KRAS mutations and predicting EGFR mutations. And also our model can keep learning from more data. So while adding more and more data, we can actually improve the performance of the model, versus supervised models are tied to data that's available for that task. Another thing that we wanted to do is we want to understand what this embedding is that we are learning, this lower dimensional space. So we extracted the vector that represents the smallest and the largest lesions, and then we try to shrink large lesions or grow small lesions. So these are images of lung cancer patients with average size. So now we can, using sort of this difference factor, we can now shrink them or make them bigger. So this was just a way of also testing whether these models can be used for generating synthetic data, perhaps in the future, by manipulating the size of these lesions. So I want to remind you that this model was not trained in this way, so we just wanted to see if we could manipulate size in this manner. So to summarize this work, so we have developed variational autoencoders for CT lung images, and we have done similar work for brain MRI and for liver CT. And the idea is really to represent these data in an unsupervised way or using self-supervised learning to then use for downstream tasks. We also saw that these unsupervised deep learning features perform similarly to radiomics, and that also the combination of radiomics and deep learning features could perhaps be synergistic. And this embedding seems to capture the lesion itself and also the size of the lesion. And now ongoing work in radiology is to develop vision transformer models to potentially replace these variational autoencoders. So let me now move on to the last part, where I want to show you a multimodal data fusion effort that we have done in glioblastoma. So I've talked already a lot about it, also in the intro. Essentially, we want to look at all these different streams of data, clinical data, molecular data, imaging data, and bring them together in a machine learning model. And here we use data from multiple cohorts, pediatric and adult cohorts in the brain tumor setting. So we used adult glioma from the Cancer Genome Atlas, and then pediatric glioma from the Kids First project. And for each data set, we had histopathology and gene expression data. And then we also had two more rare pediatric brain tumor types, medulloblastoma and ependymoma. So this here is a visualization of all the cohorts that we had. So we had, here is all the adult data. So we had low-grade glioma and glioblastoma data from the Cancer Genome Atlas. Then we had the validation data set from CPTAC, also including both histopathology and RNA-seq data. And then we had all these pediatric cohorts, the low grades and high grades, and then also two more rare tumor types, ependymoma and medulloblastoma. So first, we used very standard pre-processing for these data sets. So for the histopathology images and for gene expression data. And then here I'm going to describe the different models that we use to extract features from these images and from the RNA-seq profiles. So for the image data, we again used ResNet to extract features. And we used a very standard multilayer perceptron for the RNA sequencing data. And then the prediction that we are doing is overall survival. So we want to predict prognosis of these patients using the Cox loss. First, we wanted to look at the baseline model. So we want to know how each of these different modalities predicting survival. And we use two metrics, the concordance index and the BMIR score, to evaluate these models. The concordance index is essentially a relative metric that's bounded between 0 and 1, and it essentially gives an idea of how well we can distinguish two individual patients. So very similar to ROC, area under the ROC curve. And then the BMIR score is an error metric. So the BMIR score we want to minimize. So we want to have the lowest possible BMIR score. But that's an absolute metric. So what you can see here is that the expression data seems to be on its own predictive of survival in both pediatric and adult glioma. So now we want to develop some fusion methods, bringing these different data sets together to make predictions. So first, we wanted to use feature fusion or early fusion. That means that we're going to concatenate the feature vectors of the different data modalities. So we have a ResNet, for example, that extract features for the images, and then we have the multi-layer perceptron for the RNA sequencing data. And then we concatenate these features and make a prediction. Then late fusion is the opposite. Now we are going to develop models on each data modality separately, make a prediction, and then integrate those predictions. And then finally, joint fusion is a model where all features and parameters are trained end-to-end, and the model is essentially learning the parameters of the ResNet and the multi-layer perceptron at the same time for both modalities.
Do you have a prediction? Did you have an hypothesis as which would do better?
I would say, I was just going to say also that the joint fusion, because it's computationally more expensive, that this model probably would not do as well.
Excuse me, did you add any EHR data or it's only based on?
We just had some demographics and like age, sex, and so on. So we didn't really have a lot of, yeah. So here I'm showing the results for adult gliomas. And as you can see, the fusion models did better than the single modality models. Similarly, for the pediatric cohort, we were able to show that early, late, and joint fusion perform better than the single modality models. And we also looked at some survival curves to make that more clear. So these are the curves for single modality, so for histopathology and for gene expression data. And here are the ones for the fusion methods. Then we also looked at another way of evaluating these different models. And in the validation set, and both in the validation set and the test set, you can see that the fusion models seem to statistically perform better compared to the single modality models. And one thing that we also noticed that it seems that the variability across multiple repeats of the model, the training and testing procedure was also lower in the fusion models. Then this is a validation in external cohort. So we also tried these models in the CPTAC cohort. And here we show that early fusion seemed to be the best model. Then finally, I also mentioned these two more rare brain tumor subtypes, ependymoma and medulloblastoma, with roughly 40 and 60 samples each. So you can imagine that it's almost impossible to train a multimodal model in this fashion on these cohorts. So we wanted to test, can we learn these models to these more rare pediatric disease areas? And this is what I'm showing you here. So these are the results of the fusion models and the single modality models for ependymoma and medulloblastoma. And in ependymoma, the joint fusion models seem to perform the best versus early fusion in medulloblastoma. Then one other thing that we looked at is interpretation of these models. So these are, of course, more complicated models because we have multiple modalities. And because we're using deep learning models, they're not necessarily easily interpretable. So one way of looking at interpretation was we used, again, one of these models I mentioned before, HooverNet, to identify the cell types. And then we looked at the areas of the image that are most predictive of survival and how it overlaps with cell types. And this is what I'm showing you here. So on the top is the bad survival images or example images, and at the bottom are good survival images. And here you can see a difference in the cell distribution. So we found that, not surprisingly, that the amount of tumor cells was predictive, and then the amount of lymphocytes was predictive of good survival. Also, we can look at the interpretation of the RNA model. We don't want to look at single genes because that's very hard to interpret. So we look at pathways, and we want to see which pathways are predictive of poor prognosis versus good prognosis. So these are essentially SHAP values that I'm showing you here, where distributions on the right are correlated with poor prognosis versus on the left with good prognosis. And what we found was that in the joint model, a number of pathways were involved in poor prognosis, which are related to cell cycle and RAS pathways, which are shown here. And then a number of pathways were enriched in a tumor suppressor gene called RUNX3, which are related to good prognosis. And these were essentially ways of indirectly interpreting the model and also validating the model that the joint or the fusion model essentially correlated more with known biology in glioblastoma versus the RNA-only model. This is just an example of how we develop multimodal data fusion frameworks to predict survival. I also showed you some examples of transfer learning to more rare disease areas with these multimodal models. And so we did some work in terms of interpreting these models. So I hope I give you some examples today of the multimodal work that we do in the lab, and how we can develop models that are biomarkers that are reflected in medical images, and how we can use these to study clinical outcomes. And a promising application is non-invasive precision medicine. And also we are very interested in using imaging as a biomarker for treatment decisions and treatment response. These are the people in the lab who did actually all this work and are funding, and happy to answer any further questions.
Question about the challenge you mentioned earlier, like in the last example you showed, like how did you handle missing modalities?
So in that particular case, we only worked with the data where all modalities were available. Yeah, but of course now with these other methods, we could extend our work and generate synthetic data for any patient where we have data missing. Yeah.
I noticed that you have been using ResNet to extract features. Do you think it worth it to use image segmentation method to extract features and then manually curate them as features? Do you think it will?
So I'm not sure this answers your question, but so we used ResNet a lot in the past, but now in the past 6 months or so, 6-9 months, there have been a lot of foundation models in digital pathology and other medical image modalities. So we are switching to sort of this more customized, let's say, feature extractors and showing better results. So using sort of more, as I said, customized feature extractors, it gives us better results now. Does that answer your question?
Yeah, somewhat, yes. Thank you.
Other question?
Olivier, I didn't realize you're a mathematician, so I'm going to ask an esoteric question. We've been reading papers of people saying now they have computational models for epithelial to mesenchymal transitions with partial differential equations and Markov models. And then someone reported a whole set of virtual cells that they have, and I think they were at Stanford, they presented at Chan Zuckerberg and blew away a bunch of people. For all I know, they're from this room, in this room. anyway, should we have faith in those kinds of mathematical models? I don't know how the virtual cells are constructed, but the other ones you get the aboutness.
So this is a future interest of ours, is to link this work with mathematical models.
Okay.
So we are working, we have some preliminary work already on modeling breathing and lung and elasticity in breast tumors using partial differential equations. So I think bringing these models, these mathematical models together with deep learning, I think that's sort of the direction we want to go. And one of the concepts that we want to implement is the concept of a digital twin for a patient, maybe for a tumor, or for an organ system, and so on. So this is definitely something that we want to incorporate.
So you have some faith in that direction?
For sure, yeah.
Why don't you get in, just as time, get in a last comment yourself, and if people can approach you directly for maybe questions afterwards.
Of course, yeah.
Maybe a final word from Dr. Gevaert.
Yeah, let me know if you have any questions. I don't have anything else to add, yeah.
That works. Thank you, Dr. Gevaert.